Now there are a little bit of difference between the ABO system and the RH system in terms of the antibodies that you make. Right? So if you are a type A person, A negative, let's say, right? One more time, A negative, then you would have A and then nothing else, right? So you will be able to make anti-B and you will be able to make anti-RH. So it turns out, for reasons we're not going to go into details about, when the A negative person is born, they will already have the anti-B antibody in their system. However, the RH antibodies are not going to be produced unless the person becomes sensitized. Okay, and I'll explain what that means later. Another example of this is would be like contracting a disease. Okay, so when you're born, you do not have antibodies against chickenpox, for example. You will only make those anti-chickenpox antibodies after you've contracted chickenpox, right? So in a similar sense, the negative person will not make the anti-RH antibodies unless they are exposed to RH positive blood. Okay? So this is not the case. This is not the case for the ABO system. If you're type A, you're born with the anti-B antibodies. If you're type B, you're born with the anti-A antibodies. However, if you are RH negative, you will only make the antibodies after you have become sensitized. Okay, so sensitized means you are exposed to positive blood. So an RH negative person is sensitized if they are exposed to RH positive RBC. Okay, positive people don't get sensitized for RH. If you already have RH positive blood in your system, you are never going to make anti-RH antibodies, right? You are not going to make antibodies to attack your own self, right? So only negative people can become sensitized. So how can a person get sensitized? Well, one way that a person can get sensitized is blood transfusion. If RH positive RBC is given to RH negative person. Okay, now here is where things get a little bit tricky. If you are a negative person, you are going to be okay to receive positive blood for one time. Okay, it's like your like one free try or something. Okay, that's because if you are negative, you haven't made those anti-RH antibodies yet. So when the RH positive RBC goes into your system, you will not reject it the first time. Okay, so you get that free transfusion, but afterwards you will become sensitized. Once you've become sensitized, you will start to make the antibodies against the RH. So the second time they try to give you RH positive blood, you will reject it. You will undergo cross reaction, agglutination, and hemolysis, and the blood will no longer be compatible. Okay, so that's one example of how a person can become sensitized. Another example, another reason for people getting sensitized is if an RH negative mother is carries an RH positive fetus. Okay, if the baby is positive and the mother is negative, then the mother could potentially become sensitized. So let me explain this to you in a diagram. So for example, here we are going to consider a mother. The mother is going to be, let's say, negative. Okay, that means her RH antigen is guaranteed to be RH negative, and the father is going to be positive, right? And that means he is going to be RH positive. If this couple is going to have a kid, and you do the Punnett square, then 100% for sure their child will be positive slash negative. So positive from the father and negative from the mother. And this genotype will mean the child is Rh positive. Okay. So let's say this is the first pregnancy, and I'm going to have the mother's blood here, the mother's bloodstream over here, and this is the mother's blood, and over here we're gonna have the baby's blood. Okay, the baby's blood, the fetus. Okay, baby's blood. Right? And the mother and the baby is of course separated by the placenta. Okay, so the placenta, supplies the baby with nutrients and all that. The mother, remember in our scenario, the mother is Rh negative. So that's mother Rh negative, and the baby is Rh positive. So what happens is we are going to draw some blood for the mother. These are the mother's blood cell, and we're just looking at Rh. Okay, we're not going to concern with the ABO at this point. If we just look at the Rh, then there are no Rh antigens on the mother's RBC. For the baby, though, the baby is going to be Rh positive because of the positive allele from the father, and so on the surface of the baby's blood, we are going to have the Rh antigen. Okay, so the placenta is pretty good at separating the blood of the mother and the child. It's not going to let them mix together. The only time this is going to have mixing of blood is if the placenta is damaged. So that could happen, if there is some kind of accident during pregnancy or during delivery. It's quite possible that the placenta is damaged, and that will result in the baby's blood going into the mother's bloodstream. Okay, so let's say the placenta was damaged during delivery, during labor, then the baby's blood will now go into the mother's bloodstream. Okay, so now the mother is going to become sensitized, right? Remember, sensitization means a negative person, in this case the mother, is exposed to positive blood. The source of positive blood in this case is the baby's blood. Right? So her immune system is going to get triggered. She is going to make the anti-Rh antibody. But that's not going to be a problem for the first baby, okay? Because by the time the antibodies are made, the baby would be born, would be delivered, right? So it's not going to harm the baby, okay? So now, let's say the couple decided to have another child, right? So now this is the second pregnancy, okay? Second pregnancy. So what's different, between the first and the second pregnancy? Well, because the mother was sensitized by her baby the first time, she will now, in her plasma, carry anti-Rh antibodies, okay? So these are antibodies that are created to attack Rh cells. And what happens is, in the second pregnancy, the baby is positive, so some of these antibodies are actually going to cross the placenta. They're going to cross the placenta, and they're going to cross-react with the baby's RBC, and that is going to result in hemolysis in the baby's blood. Okay, so that is not a good thing to happen. So let's go back to the PowerPoint slides and see what's going on. Okay, so this is a more professional drawing, I guess, of what I have been explaining to you. Let's just go through this one more time. This is a negative Rh negative mother carrying a Rh positive fetus, and in the mother's blood, right, the cell does not have those little spiky stuff, which represent the Rh in this case, but the fetus does. Okay, and if there is hemorrhage during delivery that causes the placenta to be damaged, then the baby's blood will now go into the mother's bloodstream, sensitizing her. Okay, and once she becomes sensitized, she will make anti-Rh antibodies. But that's not going to affect the first baby, because by the time you make the antibody, which is like a week or two later, the baby is already delivered. Okay, but this will be a problem for the second pregnancy. If the second child is also Rh positive, then these antibodies will cross the placenta and attack the baby cell, causing them to rupture, basically. So if we do not prevent sensitization, then the baby is going to be born with something called hemolytic disease of the newborn, okay, or for short is HDN. Okay, so there is a longer name, erythroblastosis fetalis, but we can just call it HDN. Okay, so hemolytic disease of the newborn can vary in severity, right, depending on what blood group is incompatible. Okay, Rh incompatibility is only one example of how HDN can occur. There are so many different blood groups. Some other compatibility might be more severe than others. And the symptoms might include, as you can imagine, anemia, because the baby doesn't have enough red blood cell, right? And so they will be not able to deliver oxygen efficiently, so resulting in some form of anemia. Jaundice, okay? Remember, when you destroy your RBC, right, you are going to release the hemoglobin. Right? So you recover as much of the hemoglobin as possible. You take the, you take the protein, you take the heme, you take the iron, and whatever you cannot break down, then is going to whatever you cannot reabsorb, it's going to be converted and broken down into bilirubin. Okay, and so this bilirubin stuff is yellowish, right? So typically you get rid of it from the urine or feces, but now there is so much of it because so much of the red cells are dying in the baby. There is going to be a lot of bilirubin, and that will accumulate in the skin, causing the baby to appear to be jaundice or yellowish in color. Of course, all these extra red cells that are dying will be recycled at the liver and the spleen, so they're working extra hard, and that's why they will become enlarged, and there will be some a severe edema or swelling in some cases. Remember, in our discussion of capillary exchange, you have a lot of blood cells and proteins and solutes in the blood vessels, and all these things are important to maintain fluid in your blood vessels. If you now have a lot of red cells dying, then you don't have the ability to retain as much fluid, and so they're going to accumulate in the tissue surrounding blood vessel, resulting in edema. But like I said, the disease severity is going to vary from person to person. These are actually my daughters. This is the younger one. This is the older one. They were both born with jaundice, and what happened is my wife is type O blood, and both of my daughters are type B blood, and so what that means is my wife, being type O, she would have anti-A and anti-B antibodies in her system, right? So that there is a little bit of incompatibility there. But ABO incompatibility is much less severe compared to something like RH. So what happens is to help with the jaundice, they were put into the chambers, which gives them UV light. This is actually called Billy light, and is designed to break down the bilirubin for them. You might have heard of doctor recommendation before to put your babies in the sun if they appear to be a little bit yellow. It's the same principle. It's just that this is more controlled environment, and so the first one had the Billy light for just 24 hours, and then for the second one it was actually much more severe, because she actually didn't want to come out, right? And when the babies don't come out, you kind of have to vacuum them out. For those of you who don't know, and after you vacuum them out, there is a big bruise in the forehead, in the top of the head rather, and that bruise is basically red cells dying. So that further contributed to the elevation in bilirubin. So she was in and out of this chamber for like maybe like a week or so, but in the end, they were both fine. So what do you do for RH sensitization? Because that is much more severe than the just regular ABO incompatibility. So it turns out there is something called RhoGAM, and RhoGAM is an artificial anti-RH antibody. Okay, and we can use this to prevent sensitization. So I just want to emphasize that the RhoGAM does not cure sensitization. Okay, meaning if you are already sensitized in your first pregnancy, giving you RhoGAM is not going to reverse that. It's not going to undo it. Okay, this is only effective for people who have not been sensitized and you want to prevent it from happening. Okay, so what is RhoGAM? So let's go back to the to our note here to try to explain the situation to you. Okay, so let's just erase these things. Let's go back to the first pregnancy. Okay, so now the woman is pregnant for the first time, and based on blood work, the doctor see that the mother is negative, and then the father is positive. So usually we don't really test for the baby's blood type. Okay, it's much more straightforward and easier to test for the father's blood type. So knowing that the mother is negative and the father is positive, right, then they could know that there is a chance that the baby is going to be positive. Right? I say there could be a chance because you know, we make up this case, right? We say the father is positive, right? But it's also possible for the father to be positive / negative. In that case, then there's only 50% chance that the child is going to be positive, right? But regardless, if the father is positive and the mother is negative, then they're going to recommend you having a RhoGAM. So RhoGAM are these artificial antibodies. Okay, so I'm going to draw some here. So there is an artificial antibody against Rh. There is an other one. Okay, so the mother will receive the shot sometime during pregnancy, 28 weeks of pregnancy. Right? The whole pregnancy is usually about, like, 40 weeks or so. So the 28 weeks of pregnancy, you're going to receive one shot, and 72 hours after childbirth, you're going to receive another shot just to be sure. So how does it work? Okay, so now the mother has the RhoGAM, the antibody, in the system. Let's say the placenta was damaged again during delivery, so the baby's RBC is going to go into the mother's bloodstream. Okay, so there is the baby's RBC with the Rh on them. So normally this would trigger the mother's immune system, right, and cause her to become sensitized. But because we gave the mother the RhoGAM, the antibody here is actually going to destroy this fetal RBC before her immune system is triggered. Okay, so the RhoGAM prevents sensitization by destroying all the positive cells in the mother before it triggers her immune system. So a question you might have is, how come the antibodies, the RhoGAM, does not cross the placenta and attack the baby's RBC? Remember, these RhoGAMs are artificial. They are created in a lab. So the people who create these antibodies make them super big. Okay, so they are extra large, much larger than your regular antibodies, and so the size of these antibodies prevents them from crossing the placenta. Okay, they're too big to cross the placenta, and so if you administer the RhoGAM into the mother, the RhoGAM, these artificial antibodies, will only stay in the mother's bloodstream, and it will leave the baby unaffected. Okay, so if you are a negative mother and your spouse is positive, and you might be carrying a positive baby, then taking the RhoGAM will prevent desensitization, which is important for preventing the hemolytic disease of the newborn. So here is an example. There are 4 couples here with the kids. Which of the following could potentially create the hemolytic disease of the newborn? So go ahead, pause the video, and try that out. So let's see. All right, we know the sensitization or hemolytic disease can only occur if the mother is negative. So that's not the answer, and that's not the answer. So the mother has to be negative, and the child has to be positive. So at first glance, it looks like both of these are correct answer. But if you take a look at the father, here, the mother is negative, so genotype is negative. The father is also Rh negative, so his genotype is also negative. If both the mother and the fathers are negatives, then it's impossible for them to have a positive child, right? So this is not even going to be a possible scenario. So for that reason, the answer is C. This is something that you're going to be doing in the lab coming up later in the course. if we want to test for the blood type of a person, we can take the blood sample and mix it with different types of antibodies to see what results we get. And based on that results, we are going to be able to figure out the blood type. So this is the first blood sample, and over here in this well we have anti-A antibodies in it. Okay, second well we have anti-B antibodies, and the third well we have anti-D antibodies, which basically just means anti-Rh antibodies. So let's draw the blood cell here. If we drop it into the first well and it reacted with the antibody A, you know that there is a reaction because of these clumpings, right? Then that means the blood sample must contain A antigen. If you mix it with antibody B and there's no reaction, then that means our cell does not have any B antigen. And if you mix it with the Rh antibody and it clumps together, then that means this person will have the Rh antigen on them. And so the blood type of the first person is A positive. Okay, A positive. Okay. You should be looking at whether or not a reaction occurred, and if it occurred, then that letter is going to be on the surface of the blood cell. Okay. Let's take a look at the next one. We have our RBC here. No reaction with antibody A, so no A. Yes reaction with antibody B, so we're going to put a B here, and it reacted with the Rh, so there must be Rh as well. If you have both B and Rh on the surface of your RBC, you are B positive. If you reacted with all three, then that means you will have A, you will have B, and you will have Rh on the surface. Then that makes you AB positive. And if nothing reacted, then there is nothing on the surface of the RBC. That makes you O negative. So there are more practice in the course pack. Please go ahead and try that. Let's try a matching question here. So let's say there are 4 couples here and 4 potential babies, and you have to match them up. So to do these questions, you should just think about some of the key things that we talked about in the lecture so far. So there are 4 key things to remember for these type of questions. For example, AB people cannot have type O child, right? Type O people cannot have AB children, right? And I'm just going to add 2 more. If you have type O and another type O person, all they are going to have is type O, because it's little I for both of them. Then they could only pass on little I. As a result, the whole family is going to be type O, right? If someone is Rh negative, and you have another person that's Rh negative, then all their kids will be Rh negative, right? If you're negative, that means you're negative, and here negative. So you can only pass on negative, right? So all the kids in the family will be negative. So keeping in mind these 4 things, it should help us do questions like this one. So first, we want to find couples with AB or with O blood group in them. Okay, so let's take a look at the first couple. Yeah, so we have a AB mother. So AB people cannot have O children. So based on this fact, I know this is not their kid, and I know this is not their kid, right? All right. And then we look at their Rh group. The mother is negative and the father is negative. So 2 negative people can only have negative kids. So that means this is not their kid. This one is positive, right? It cannot be their kid. So for that reason, the kid that belongs to them is baby L. Okay, B negative. That's their child. Okay, so this baby is gone. Is taken. Let's take a look at another one. And again, we want to search for parents with either AB blood group or O blood group to work with first. So I'm going to look at the couple number 4. Okay, couple number 4, the father is type O. So type O individuals cannot have AB children. So automatically, because the father is type O, I know this is not their kid. I know that's not their kid. So there are only 2 other options, either baby K or baby M. But they are both negatives here, right? Which means if you're both negative, you can only make negative children. So given these 2 options, it's going to be baby M. baby M is going to be theirs. Okay, so baby M and baby L are taken. And now it doesn't really matter which one you look at, I guess. We will look at couple number 3. Okay, B positive and B positive. Okay, so if you are B positive, then that means the mother does not have an IA allele, and the father does not have an IA allele either. So that means they cannot possibly produce a type AB baby. So that means baby K is theirs. And finally, process of elimination, baby J belongs to couple number 2. Let's do another one from the workbook. This is question 11 in the lecture 2 study workbook study guide. Here we have four parents and 4 babies to match, and we can use the same logic that we did for the last question. We want to start with AB parents or O parents. So here we have a O negative mother and an O negative father. 2 O individuals can only have type O babies, and 2 negative parents can only have negative babies, which means this must be their baby. Okay, baby L. Next, we have AB negative mother and O negative father. So AB people cannot have O child, so that's not theirs. O parents cannot have AB babies, so that's not theirs, which means baby H. Baby H is going to be theirs. Okay, baby H. Okay, baby H. So 2 babies left, 2 parents left. O negative and B positive. Right? If you're O negative, you cannot have AB baby, and there are only 2 babies left. That means this one is yours. Okay, you cannot have AB. Okay, so you see, you don't even have to do a Punnett square for these questions. You just gotta have to logic your way through it. And finally, right, process of elimination, baby J is going to be theirs. Okay, so there is another one here. You can try it on your own, and you can compare the answers to the answer key I posted later on. I do want to go back here and talk about blood transfusion with the Rh blood group as well. So by now you should know about the ABO compatibility. I'm going to draw it out for you again. O can be given to all the other 3 blood groups, and AB can receive from all the other 3 blood groups. So that creates a chart like that. Okay, and for the Rh, I didn't say it explicitly, but you should know by now that only negative can be given to positive. Okay, if you give positive to negative people, then they are going to get sensitized. So I know I said that, you know, negative people can get like one free getaway transfusion, right, from a positive person, right, because they haven't been sensitized yet. But in the test questions, if I ask you, can positive be given to negative, then you know the answer is generally no. Okay, we want to avoid sensitization wherever possible. So based on that, let's take a look at here. If you are a positive person, who can you receive blood from? So if you're type A, you can take a look at here. If you're type A, right, then you can receive from other A. You can receive from other A, and you can receive from other O. Okay, those are the people you can receive from. And if you're positive, you can receive it from either positive people, or you can receive it from negative people. Right? Positive can receive from negative. Who can you donate to? Right? If you type A, you can donate to other A, and you can donate to AB. But because you're positive, you can only be donating it to other positive people. Okay? You cannot give your blood to negative people. You're going to sensitize them, and we don't want it. Okay? So go ahead, try it with B. I'm going to try the AB with you. Okay? If you're AB positive, who can you receive from? Well, if you're AB positive, you are really lucky, because AB can receive from everybody. So you can receive from A, you can receive from B, you can receive from O, and you can receive from AB. And if you're positive, you can kind of double dip. You can receive from the A people, positive people, and you can receive from the negative people. Okay? So who can you donate to? Only other AB, no one else. Okay? Because AB can give to anyone other than the same blood type, and positive can only give to positive. Okay? Let's try the B negative here. Okay? If you're B negative, who can you receive from? Okay? B can receive from other B's and O's. Okay? But if you're negative, you can only receive from negative but not positive because it will sensitize you. Who can you donate to? Well, B can give to other B as well as ABs. So B and AB. And if you're negative, you can give to other negatives and you can give to positive as well. Okay? So try this out and see you know, if you can figure out the entire transfusion chart. Now, this is another chart that you should definitely try. I'll do a few examples with you here. So we have the donors here and we have the recipient here, and we want to figure out if it's safe or not to do the transfusion. So A positive can give to A positive because it's the same blood type. B positive to A positive, is that okay? So B to A, can B be given to A? Well, the answer is no. So you would know that either from the transfusion chart here, or better, you would know that this is not okay because the recipient will have anti-B antibodies, and that's going to attack the incoming RBC. Okay, so this is not safe. It doesn't matter that positive and positive are compatible. Okay, there are two things you have to check here. You have to check for ABO compatibility and you have to check for RH compatibility. Both blood group has to be compatible for the transfusion to be safe. Okay, if either one of them is not compatible, you can't do it at all. Let's take a look at this one. Right? O negative to A positive. Okay, O negative to A positive. Can O give to A? If you check the transfusion chart, the answer is yes. Okay, can negative give to positive? Yes. So because both blood group are compatible, this is going to be safe. Okay. Let me see if there is another example here. Okay, B positive. Okay, B positive to AB negative. Is it safe? B positive to AB negative. Is it safe? So first you check, can B be given to AB? Right? B to AB is okay. If you check the transfusion chart, that's okay. But can positive give to negative? No. You will sensitize the person. So this is not safe. Okay. So finish the chart and then check the answers later. But the point here is you have to check for both blood group before you decide whether or not this is going to be a compatible transfusion.